nuclear organization and genome function. Eukaryotic cells face the challenge of compactly packaging the genome within the cell nucleus while maintaining accessibility for dynamic processes like transcription and DNA repair. Chromosomes during interphase are organized into distinct territories influencing potential interactions and determining the genomic locus, nuclear environment. The nucleus contains subnuclear to nuclear bodies, dynamically regulating various nuclear processes for greater efficiency. Molecular organization. DNA, the nucleus, is meticulously packaged in the chromatin, which condenses into chromosomes during cell division. Chromatin consists of nucleosomes forming a wedge on a string, structure that compacts DNA. New chromatin is genetically active, while heterochromatin, more condensed, is often inactive. Throughout a cell's life, chromatin fibers change form with dispersed chromatin during interphase and compact chromosomes in metaphase during cell division. The number of chromosomes varies among species but doesn't correlate with complexity. The nuclear envelope is a regulated membrane barrier that separates the nucleus from the cytoplasm. It plays a crucial role in chromatin organization and gene regulation. During mitosis, the NA undergoes precise assembly and disassembly posing challenges in cell division. The nuclear envelope formation involves targeting membrane to chromatin, membrane fusion, and the incorporation of nuclear complexes or NPCs. In vitro studies like xenopause egg extracts provide insight into de novo NA formation around DNA templates. The nuclear envelope or NA growth undergoes growth after initial formation, requiring the addition of membrane and new nuclear core complexes or NPCs. Unlike the initial formation, growth involves distinct mechanisms such as adding membrane to the outer nuclear membrane or ONM and redistributing it to maintain regular ONM and inner nuclear membrane spacing. Structure of nuclear pore complex. Nuclear pores are minute openings connecting the nucleoplast and cytoplasm, functioning as biological valves. Each nuclear pore complex is complex, comprising about 3,000 to 4,000 pores per cell, allowing selective movement of chemicals.
Importance of regulation of gene expression. The regulation of gene expression is of paramount importance for the proper functioning of cells and organisms. It ensures the precise control of routine synthesis, allowing cells to respond dynamically to internal and external signals. Coordinated gene regulation is exemplified in embryo development, where metabolic, psychological, and environmental factors govern gene expression. In E. coli, the utilization of lactose as an energy source triggered the synthesis of beta-galactosidase, facilitating lactose hydrolysis. Conversely, in the absence of lactose, beta-galactosidase synthesis is unnecessary, showcasing a responsive and energy efficient regulatory mechanism. Prokaryotic gene and regulation. In prokaryotes, gene regulation primarily occurs during transcription initiation. Accessory protein mutilate RNA polymerase activity at the promoter, acting as activators or repressors. Operators located the promoters interact with repressors proteins, influencing promoter accessibility. The lack operon exemplifies this, showcasing specific operators and repressors of individual operons. The lack operon. The lack operon, focusing on lactose metabolism in bacteria, includes a regulatory gene, I, producing a repressor, 3 structural genes, Z, Y, and A, encoding enzymes for lactose utilization. In the absence of lactose, the repressor binds to operator, hindering RNA polymers activity and preventing gene transcription. Lactose acts as a inducer, disrupting the repressor-operator interaction, allowing RNA polymerase to transcribe the operon genes. This negative regulation system orchestrates lack operon genes expression based on lactose availability. This figure diagram showing at which stage in DNA mRNA protein pathway expression can be controlled. Gene on DNA, and the second primary transcript, third mRNA, and fourth nucleocytosome, and first transcriptional control, RNA processing control, RNA transport control, and fourth translation control. Prokaryotic and eukaryotic gene regulation. Prokaryotic gene expression primarily involves transcriptional control, where synthesis of protein occurs simultaneously with transcription. In eukaryotics, the transcription and translation physically separated. Gene regulation can occur at multiple stages, including transcriptional, post-transcriptional, translational, and post-translational levels, providing intricate control over protein synthesis. Expression of genes. The process of producing RNA and protein from DNA is tightly regulated in cells. This regulation observed in both unicellular and multicellular organisms conserves energy and space by selectively activating genes when needed. Efficient control prevents unnecessary energy expenditure and maintains a manageable cell size by expressing only a subset of gene in each cell. Gene regulation makes cells different. A crucial process that contributes to the diversity of cells. By selectively turning genes on or off, cells can acquire distinct identities and functions. This dynamic control ensures that each cell type expresses specific sets of genes tailored to its role within the organism, leading to cellular diversity and specialization. Franz Andreas Bayer, 1758-1840, was an Australian microscopist and botanical artist born in Wilhelmsburg, Moravia. His talent in botanical illustration emerged early under the guidance of Father Norbert Bocuse, leading to over 2,000 watercolor drawings of plants. Bayer, along with his brother Ferdinand, worked as flower painter in Vienna, and later Franz became the first botanical illustrator at the Royal Botanic Gardens, securing this position due to his exceptional talent recognized by Sir Joseph Banks. Bayer spent his life at Kew, creating intricate and detailed botanical and anatomical illustrations, suturing royalty in illustration techniques and contributing to various significant publications like delineations of exotic plants and illustrations of orchidaceous plants. His legacy resides in his meticulous and exquisite botanical artwork.